I'm Corrado Calisor. So I am happy to introduce now Eleftherios Zisis. Eleftherios is a postdoc at the Blue Brain Project. He obtained his bachelor at the National Technical University of Athens in applied physics and mathematics. And then he obtained his master, and he actually very recently got his PhD in computational neuroscience at EPFL. And his research includes the anatomical reconstruction of NGV architecture using Monte Carlo methods for simulate special point patterns. So with no further ado, Eleftherios, the stage is yours. I'm happy and looking forward to hear your seminar.
Thank you, Corrado. Please let me share my screen. So, hello everyone. I'm Eleftherios. I'm a postdoc at the Blue Brain Project. I got my PhD thesis, as Corrado said, recently, and I will present the architecture of the NGV system, how we can build such a system. So first, I will describe the motivation, then the framework to build such a system. Then I will proceed in explaining how we can build realistic morphologies using topological methods. And finally, I will describe the number of experiments I did with this system I built in order to build insights about the anatomical constraints of this organization. So the Neuronal-Glial-Vascular system consists of the interlinked neurons, glial cells, and the vasculature. And in this work, we focused in the most common type of glial cell, which is the protoplasmic astrocyte. And the protoplasmic astrocytes project processes to synapses where they wrap around and they form tripartite ensembles. And they also project perivascular processes to the vasculature where they wrap around the vessels to form astrocytic endfeet. In addition, with their morphologies, they form anatomically exclusive domains with neighboring astrocytes, and this organization ties to the entire cortical space. And there is a small overlap between these domains, where they form gap junctions in order to establish communication pathways and form in situ. And this complex organization gives rise to a plethora of biological functions. For example, astrocytes are involved in neurotransmitter recycling. They uptake glutamate from the synaptic cleft in order to avoid excitotoxic effects. They're involved in energy support. They uptake glucose from the vasculature. They convert it to lactate, and they provide it to neurons through specialized pathways. And they're also involved in water and ion homeostasis. And their function also depends on their location. Because depending on where they are, they express different receptors. And in addition, where there is local inflammation, their morphologies change, they become reactive, and this leads to pathological effects. And finally, the interlinked domains allow the redistribution of ions to remote areas. So in order to understand this complex organization and interplay, the goal of the NGV is twofold. First, build a realistic model of the geometry of the neurons, the glia, and the vasculature, and also implement functional models to simulate the behaviors. And you will hear about the simulation afterwards by Polina. So the questions behind the NGV is what are the components that we need in order to build such a system? How we can generate realistic morphologies embedded in this organization of astrocytes, and finally, how we can use this model in order to extract predictions and understand the anatomical organization. So the NGV framework uses data. You already heard about the generation of astrocytes, the reconstructed astrocytes, how there's and the meshing from R1, and then these astrocytes are skeletonized and they're used as inputs, a small subset of inputs, in order to drive the generation of a lot of astrocytes to build the model using topology methods I will explain afterwards. And also we use data from the literature in order to constrain our model. So there are two levels of detail in the NGV framework. The first is the network level, and the second one is the more microscopic degeneration of the realistic morphologies and their corresponding endfeet on the surface of the vasculature. But first, in order to make the NGV, we need the neurons and the vasculature. And the neurons are generated, the circuits of the neurons, with previous work from our lab, combined with experimentally reconstructed vasculature datasets from the lab of Bruno Weber. And these two datasets are colocalized, defining the bounding space for the NGV, where we're going to place our astrocytes. So the next step is to generate these locations of the somata of astrocytes. And in order to do that, we have to take into account their density, how many astrocytes you have across the cortical depth, and the distancing between them because of their tiling organization. And previous approaches solved the problem of reproducing locations with respect to density, but they didn't address distancing between cells. Therefore, we developed a new model where to place a cell, there is a probability that depends both of the density of the population, how many cells you have per cubic millimeter, and also a repulsion function that ensures the distancing between them. And this is on the left an example of such placement. In blue, you can see the somata of the astrocytes, and in red the vasculature. And we validated the density across the cortical depth in orange here, in reference, and make sure that it matches the literature profile. And this density gives rise to an average one, which lies within the range of the juvenile animals. And this is our target because we are building P14 rat somatosensory cortex models. And as far as the distance between astrocytes is concerned, you can validate that either with a pair correlation function or by directly measuring the nearest neighbor distance and validating against the literature results. So the next step after you have the locations of the somata is to generate the domains of astrocytes. So what do we know about the domains? We know that astrocytes grow and create domains, anatomically exclusive domains with contact spacing. And this organization causes a tiling of the cortical space. So in order to generate this partitioning of space, I use the mathematical abstraction of the domains using Voronoi tessellations to generate these bounding regions and ensure that they tile the entire space. And this will act as our bounding volumes within which we're going to grow our astrocytes. So this is an example of such partitioning of the space in microdomains. In B, we can see in more detail the edges of the domains, and in blue the somata in the center. And in C, an illustration of one domain and its neighborhood, along with randomly generated morphologies. And I validated and made sure that the distribution of the volumes lies within the correct range of juvenile animals in order to make sure that we are not going to generate either very big or very small astrocytes. So following the microdomains, the next step is to establish the connectivity of our system. And we know that astrocytes project to the vasculature and that they also incorporate as much as 60% of synapses in their domains, and that they form gap junctions with the neighbors. So this gives rise to three types of three types of connectivity. So the first is the gliovascular connectivity. So in order to calculate that, we distribute points on the skeleton of the vasculature, and then we find which ones of them are inside the domain. And according to literature constraints, we choose the number of endfeet per astrocyte. In a similar manner, we calculate the neuroglial connectivity, finding the synapses that are within the bounding regions, the microdomains of the astrocytes, and selecting a subset that corresponds to the 60% that we encounter in literature. And finally, the glial connectivity is calculated from the geometrical intersections between astrocytes and their neighbors. So in figure A, you can see the projections from an example of the projection from astrocytes to the vasculature. In B, we can see the gap junctions that are established between neighboring astrocytes in green. And finally, in C, we can see the synapses that are included in one such domain. And we made sure to constrain the model as much as possible given the available data. So we validate against, for example, the perivascular connections per astrocyte, and also the distance from each astrocyte to the closest neuron. And we found that it lies within the correct range as found in the literature. So after we have generated the network level information, the next step is to go deeper and generate in detail our astrocytic morphologies. And this process has two steps. First, synthesizing the morphologies, and then reconstructing the geometry of the endfeet on the surface of the vasculature. So I will first describe the endfeet reconstruction to leave for the end the synthesis. So for the endfeet, we know that they oppose on the vasculature. They don't overlap with the neighboring endfeet, and finally that provide the coverage of the entire vasculature. And in order to model that, I developed an algorithm that starts from the connections that have been established at the network level, and then we propagate waves from these connections along the surface of the vasculature, which then they can converge to neighboring waves and form this kind of tiling behavior on the surface of the vasculature. And then these areas are pruned in order to match the distribution of area that we found from the vasculature, from the literature. And here we can see an example of such the result of such an algorithm. In blue are the endfeet generated from the vasculature, and in red is the vasculature, of course. And then also validated that the distribution is properly constrained from the data that I have extracted from the work of Kali. And when we first ran the algorithm, we had a coverage of approximately 90%, and then pruning it resulted to a coverage which is approximately 57%, which is in accordance to the reported values for cryofixated tissue. So what is left is to generate the morphologies of astrocytes embedded in space. And in order to do that, we have to take into account multiple things. First, we need to reproduce the complex branching patterns of astrocytes. In order to do that, we use a topological approach where the branching pattern is decomposed in a topological persistence barcode. And then we use these barcodes to generate new astrocytes, to grow new astrocytes matching this topology. And we also take into account that astrocytes extend processes to the synapses in the neighborhood, in the local environment, and they also grow radially processes that then ramify. So in order to simulate this behavior, we use an approach called space colonization, where basically when you grow a process, the process is influenced from the local environment of, in this case, synapses, and grows covering the entire space. So this combination of topological growing and the space colonization is called topological colonization. And this is an example of such a circuit with a synthetic astrocyte. In B are 6 examples of the circuit, and in C, a closer one astrocyte with its corresponding endfeet. And we validated the morphometries with respect to the experimental measurements from the dataset of Corrado, and also the topology by calculating the topological distance between synthetic astrocytes and the biological ones. And within biological population, we found that the distance of the synthetic to the biological ones is smaller than the biological ones have within the population. So having made the circuit, so this block of tissue that has neurons, astrocytes, and the vasculature, the next step was to do experiments to extract predictions about how it's organized. So the first experiment I performed was to calculate the spatial density of different components, such as the large vessels, the capillaries, the astrocytic somata, and the endfeet contacts. And I found that the endfeet contacts on the vasculature are mainly correlated with the density of the somata, not so much of the geometry of the vasculature itself. And it's interesting because it means that the space filling geometry of the vasculature does not lead to clustering of endfeet. And as a matter of fact, if we take a cross section at some height and plot the endfeet contacts overlaid by the space filling vasculature, we see that there is no preferential clustering in this organization, which means that there is a uniform provision of tropic support across the lateral direction, but changes across across the cortical depth. I then extracted the numbers, for example, the wiring of neurons, astrocytes, and the vasculature, and I found there is an order of magnitude difference between the wiring of neurons, astrocytes, and the vasculature, which is also found in the literature. And this is a result that is emerging without an extra constraint because of the geometry of its anatomical components. And this is because neurons span multiple layers, have higher density than astrocytes. Astrocytes are more localized and to lower density. And finally, the vasculature has is space filling but has low occupancy. And the final experiment was to generate 100 NGV circuits where I increased the number of astrocytes, and I measured the number of endfeet in this space that was unchanged, and all the parameters remained the same apart from the density. So I found that the total number of endfeet decreased sublinearly instead of linearly increasing. And then when I took a closer look, I found that the endfeet per astrocyte decreased as the number of astrocytes increased. And in addition, as the density increased and the domains became smaller and the packing led the astrocytes closer to the vasculature, that occluded the access of neighboring astrocytes to the vascular sites to form endfeet. And this is interesting because it could be an implication that happens in case when you have local proliferation of astrocytes. So in summary, I presented how we built the NGV digital reconstruction of neurons, protoplasmic astrocytes, and the vasculature for the P14 rat neocortex, where we placed astrocytes according to their density, cortical density, and dispersion. And then we use these locations and size of the somata in order to build mathematical abstraction of the domains of astrocytes that were used as bounding regions that where we extracted the connectivity from and use them also for guiding the growth of astrocytes, and also generating the geometry of the endfeet on the surface of the vasculature. And finally, I saw that we can use the circuit in order to extract the analysis and predictions for the anatomical components of such a circuit. And I would like, before finishing, I would like to thank all the people from both BBP and KAUST. So thank you all. Questions?
Thanks a lot, Eleftherios. So I think we have time for one quick question. So if anyone from the audience has something to ask. Well, if it's not the case, for benefit of time, I would probably leave for the discussion after the end of this session. So with no further ado, I introduce you Polina Shichkova. Polina Shichkova is a PhD student at Blue Brain Project at BBP in the molecular systems group. So she's working closely with Dan Keller and with Jay Coggan, and over the years we have seen a lot of her very impressive work during our gathering together when we could. So I will leave you the stage, and I'm looking forward to hear your seminar.
Thank you for the introduction. So I'm so grateful for this opportunity to tell you about my work on the reconstruction and simulation of the neocortical energy metabolism. So first of all, what do we study? The brain uses a lot of energy, but it's still incredibly efficient. And to understand how the brain supports its high energetic needs and what are the implications and consequences of the underlying biological processes, we started with a detailed reconstruction and simulation of the Neuronal-Glial-Vascular unit, which is an ensemble of the neuron, astrocyte, and piece of vasculature, and all the underlying biochemical processes inside. And then we provided every neuron in the neocortical column with its specific version of the NGV unit. So why is it important to look into the energy metabolism aspect of the brain function? Well, first of all, energy metabolism can constrain neuronal signaling. Then it expands our fundamental understanding of the biological basis of the brain function. And last, but extremely important, is that the malfunction of metabolism is involved in many brain disorders and aging. So how do we develop the models? We use iterative bottom-up workflow that allows us to start from the simple model and reconstruct it, gradually expand its complexity and include all the biochemical pathways of our interest. And at the first step, we reconstruct metabolism as a system of equations, and we currently have 168 differential equations in the model. And then we simulate the system, and next, we use various data for validation, which includes but not limited to the both signal data, oxygen, glucose index, NADH, NAD ratios. And then we refine the model and repeat the previous steps unless we are satisfied with the results. And we perform analysis and make predictions for different questions where no consensus is yet achieved in the field. So let's now have a closer look at the first step in the workflow, the reconstruction. So we start with literature curation, and we assemble a database of the rate of chemical reactions and transport, the kinetic parameters for those equations, and the concentrations of metabolites and enzymes. And then we build a system of equations to describe the change of metabolite concentrations in neuronal and astrocytic cytosol and mitochondria, as well as extracellular space. And we also simulate the blood flow that provides oxygen, glucose, and lactate. And quite often, the data that comes from different literature sources may be obtained under different experimental conditions. So we have a range of values for, instead of just one precise number, for many parameters in the model and many variables. So we have to use constraint parameter optimization to solve the data discrepancies. And next, as we had this unitary model of metabolism developed, we want to bring it to the neocortical column and scale up to many NGV units connected together. And how do we do this? Well, the ATP-sensitive neural electrophysiology models allow us to bring metabolism to the neocortical column so that we provide every neuron microcircuit with its relevant specific version of the NGV unit metabolism model. And currents and concentrations changes of sodium, potassium, calcium, ATP, ADP, and also membrane potential and synaptic releases of glutamate and GABA serve us to build the interplay between energy demand by the signaling and supply by the metabolism. So after we put in place these elements, we want to run the simulations. And how do we do this? The problem is that different timescales for metabolism and electrophysiology require parallel simulations with coupling or synchronization at defined time points, where the ATP demand resulting from the electrical activity simulated with neuron simulator is given to the simulation of the metabolism and the blood flow model, which is implemented in Julia language. And this simulation of metabolism provides specific updated metabolite concentrations as a feedback to the next simulation, next time step to be simulated with electrical activity simulations. So this is kind of like feedback loop between demand and supply of the energy. And now let's have a look at what we see. As soon as we develop the model and simulation framework, we examined the activity of the neocortical column with metabolism. And indeed, both spontaneous activity and activity driven by the thalamic input are affected by energy metabolism and the ATP levels. And we observe higher firing rate in the simulation with enabled metabolic feedback. By the thalamic input driven simulation with metabolic feedback, the response to stimuli actually less prominent, which may potentially be expanded by preceding spontaneous activity of increased frequency as compared with the simulation where no metabolic feedback was supplied. So next, we wanted to have a quantitative evaluation of the network activity under different ATP levels, so that we use the voltage traces to calculate Pearson correlation coefficient for 10,000 pairs of neurons. There we first perform melting to reduce noise. So you can see the distributions of the Pearson correlation coefficient for simulations with different ATP levels, the figure on the left. And we had 5 simulation setups. First, shown in the light purple color here, is a setup without metabolic feedback. So neurons firing just use up all the ATP they have, and you see the low correlations here. And the corresponding ATP concentrations are given in the bottom histogram for all the setups. So then we supplied constant concentrations of 0.1 or 1.2 or 5 millimolar ATP at every coupling step of 500 milliseconds. And we saw how it changes the correlations in the network activity. And the fourth simulation was actually the most in vivo closed state, where we have a supply of ATP from metabolism with a baseline ATP levels of 2.2 millimolar, but the real supply of ATP is calculated, simulated in the metabolism model and given to the simulation of the electrical activity. So here it is still work in progress, but what is already interesting is that 2 questions arise. First of all, which ATP levels are actually required to support neuronal signaling and the network activity? And what are the other roles of ATP for which we need around 1 or 2 millimolar concentration, as reported by many studies? So for example, there was a hydrodrop ATP role suggested in Patel Science 2017 paper. So it's really interesting as we see quite robust behavior of the network in a wide range of ATP concentration. What is the ATP concentration needed to support signaling? And the second question is whether depleted concentration in the setup where no metabolic feedback was given is actually still relevant for the healthy state network in a short-term simulation, or is it already the diseased state process? So next, we looked at the difference in ATP consumption at the network scale. And on the left, you see the moving line, which shows the time frame along the thalamic stimuli, which is based on the in vivo pattern data. And on the right, you see how the neurons respond in terms of ATP consumption to those stimuli. And we see the laminar organization in ATP consumption distribution, where layers 2 to 4 were the fastest energy consumers. And we also see that when the thalamic stimuli are highest, then after that, we have quite high consumption of the ATP. And then the differences in laminar kind of organization and ATP consumption can probably partially, at least for layer 4, explained by the thalamic innervation in this study. So next, we address the energy constraints that metabolism poses on circuit activity. And in these 2 simulations, we injected the same current of 0.1 nanoamperes to all excitatory cells for 500 milliseconds to make the neurons really firing a lot. And this protocol is not relevant to in vivo conditions, but it's important for analysis purposes. And we can observe that higher firing rate is in the setup with higher ATP levels, and it's also preserved for longer than in the setup where we give a feedback lower ATP levels. But further, we look at the energy consumption at synaptic processes in the same setup. So we could see the high number of synaptic release events requires high ATP levels due to the metabolic cost of synaptic processes, and decreased ATP levels are actually causing or associated with the decreased number of release events per neuron. And we are looking here the release events per presynaptic neuron and also at the received events per postsynaptic neuron. So next, we extracted 45 descriptive features from the voltage traces in different setups and analyzed how ATP affects characteristics of the neural activity of individual neurons of different types. And we could see that many features are affected by changes of ATP levels, but these effects are also different in different neuronal types. So the effects, for example, the features that we analyzed are related to those describing the action potential, width, amplitude, or duration, also after hyperpolarization, depth, and other features, with some more examples given here. So next question we addressed was about differences in the ATP consumption by excitatory and inhibitory neurons. And we saw the decrease in the ATP affects the range of electrophysiological features in those neurons, in the sense that we see decreased variance when the ATP levels are decreased. But overall, ATP consumption was quite similar in excitatory and inhibitory neurons. And further, the changes in this baseline baseline ATP concentrations in the physiological range would only lead to these moderate differences in features describing neuronal firing. But more interestingly is in the plot on the right that you could see the difference in the skewness of ATP levels distributions in low and high baseline ATP setups. So this actually supports the previous findings of the Lemasson from 2014 paper from Neuron. There it was shown that reduced ATP availability actually increases the metabolic cost of action potential, and then it was called as like neuron entering the deadly loop of energy consumption. So last, we use the extracted electrophysiological features extracted from the voltage traces to perform the dimensionality reduction with t-distributed stochastic neighborhood embedding. And this allows us to identify the signature characteristics that are responsible for the differences in ATP consumption. And again, we see correlations between the ATP consumption, like it's on the left plot, and they are related to the difference in the layers and difference in cell types. So with this, I'd like to sum up what we did so far. We reconstruct and simulate the NGV unit and the energy metabolism driven by the neocortical column and giving constraints to this signaling activity. The physiological levels of ATP are required for the network activity, and the low ATP levels can actually be associated with some synaptic failures, for example. And ATP consumption in column driven by thalamic innervation shows laminar organization where layers 2 to 4 show the highest energy use. And the combination of electrophysiological features extracted from the voltage traces explain the differences in ATP consumption by neurons of different layers and types. And here are my thank you to all the Blue Brain and KAUST people involved in this exciting project. And now I'd like to stop, and I'll be happy to answer your questions.
Polina, thank you very much. We have time for questions. So I would ask the audience if someone would like to start the discussion. Of course, we have a question from Professor Magistretti. I was sure about it, Pierre. So go ahead.
Thank you, Polina. And this is obviously not too new to me. I mean, even though some of the latest data I wasn't fully aware. I mean, it's really now coming together very nicely, I have to say. I remember we in our exchanges with the two groups had many discussions, and now it's really coming out as a coherent story. I'm not so surprised, I guess, from the physiology, you know, that layer 2 and 4 and 2 to 4 neurons, you know, are the ones that in a way, I mean, ATP is highest, meaning I assume that they would be those that use most energy. Still, the question which I'm not entirely clear about, and maybe I missed something, is on the one hand, you say that there is no difference between inhibitory and excitatory neurons in terms of consumption, which is interesting. I mean, this has been a very long ongoing debate, you know, whether inhibition costs more than excitation. And so, I mean, that can bring another viewpoint. But then you say that there are differences in consumption depending on the neuronal type. So that there is, could you clarify what you mean with this, actually the latest?
Sure. So here they look at the several aspects. One aspect is a difference in the electrophysiological features extracted from different types of neurons under different ATP baseline levels or ATP conditions. And the other questions is the actual consumption rate. So if you look overall at like a populations of neurons, when we give a thalamic input, then we don't really see much significant difference in that. But I need to do the formal analysis for this. I was not yet the time to perform this formal analysis to compare statistically the differences. Yeah, and then like the last part, when I do the prediction or dimensionality reduction for visualization purposes, and then I color the resulting plots by different ATP levels and types, there is indeed some difference between layers and less difference between the different types of neurons. And yeah, it's all requiring quantitative evaluation of those differences, but just by looking at those graphs, I see more importance of the layers here rather than like individual neuronal types. And yes, I'm aware of this debate about excitatory versus inhibitory activity, but also what is really interesting for me is that how these differences are evaluated at the network scale when you take the neurons connected together and not just one neuron model, which is given some input. So this is also an interesting question, like which inputs are given to those neurons so that they give this, get this energy used. So yeah.
Yeah, I guess, you know, as you said, I mean, you're almost done, but there is still some work. So I think it will be good to that we have, you know, in the upcoming conversations, we address, you know, kind of clarify a little bit this aspect, which is interesting per se. And I think you have the data to actually clarify it. Well, anyway, well done. Thank you.
Thank you. And I see the question on the chart.
Yeah. Okay.
On the slide 7, is the coupling bidirectional? Yes, as I remember my slide, yes. It is bidirectional. And if so, what's the criterion which integrates and exchange the information between the two coupled simulations? So maybe I can share my screen again. Let me try to do this just to show that slide. Okay. Okay. Yeah. Oops. So here the criteria or the variables are listed in the center. So you see the number of release events, the changes in concentrations of ions, their currents, and those things simulated as a demand side by electrical activity are given to the metabolism model because they have like the synchronization between those two simulations at some time points, and as it's shown in the bottom here. And then we simulate the changes of metabolite concentrations in the metabolism and blood flow model, and we can tell what should be the actually updated concentrations, and they give these concentrations as initial conditions to the next step of simulation with electrical activity simulation. So was this clear?
Thank you very much, Polina. So I think you're welcome. So I think it's time for our last speaker. Finally, it's my very, very great pleasure to introduce you Dr. Jay Coggan, who is our senior scientist. I would say Jay has has a previous long experience in single cell neurophysiology, and he's a scientist with the molecular system group at the Blue Brain Project. His job is to model brain energy metabolism in the context of NGV ensemble, and he has done some impressive work during these years of collaboration together with KAUST. So I'm happy to leave you the stage.
Okay, so I'm going to talk today about a secret language of cells, which is based on the ligand-receptor-cascade communicating through what are called molecular phase transitions. And so brains come to mind when most people think about biological intelligence. And certainly at the Blue Brain Project, we are laser focused on the reconstruction and simulation of mouse brains. But it is nonetheless important to remember that single-celled organisms have been making successful decisions for billions of years, and that the properties of mammalian brains are basically innovations built on the thoughts of cells. And so this work is motivated by the hypothesis that there are yet to be discovered information processing mechanisms in single cells, and offering a proposal for what one such a mechanism might look like, a secret language, if you will. So what are some of the theoretical requirements for a secret language? Well, first of all, we haven't measured it yet, so this is a prediction. But such a language should capture the intensity features of a stimulus. It should capture the time features and the evoked state changes inside the cell. So I think these are the three minimum requirements for an information processing network. And so the test case is the metabolism we've been working with, which is stimulated by norepinephrine at beta-adrenergic receptors, which then stimulates cyclic AMP cascade and results in the formation of pyruvate and downstream products such as lactate, which is then exported from the cell. So I'm using three different norepinephrine waveforms to test this theory. One is an amplitude modulation here, one is time modulation, and one is both, to see which features we can capture. So we'll start with some interesting properties of the ligand receptor binding design features. So here I'm passing the waveforms sets, basically, amplitude, time domain, and mixed, through a series of receptors with different affinities, with KD being the inverse of the affinity. And what we see here is that at very high affinities, the receptors are basically event detectors. There's really no difference between the waveform and the response evoked. But as we go through a series of receptors down to very low affinity, we see increased spread. And finally, at the lowest affinity, if you rescale, just because these amplitudes are low due to the low affinity, we have complete replication of the original waveform. So you can think of this as input and output. And this principle holds true for time domain and mixed domain. And of minor note is that there seems to be some sort of frequency spread, you might say, for the time domain effect. And if you come down and look at just amplitudes, again, you see the same effect, where you get complete recovery of the original signal at low affinities. So high affinity receptors are really just basically event detectors. And there are also a lot of interesting properties. You can see that there's a difference emerging between the capture of amplitude information when you have just an amplitude only signal or a mixed signal, because these should be the same. So we look further at that and we see that if you plot the peak of the receptor activation against the different receptor KDs for every tau, which is going to be the shape of the stimulus, you can see a spread in the response. For the time domain only signal, you naturally don't see much difference because it's mostly a time domain change. And for the mixture, you see that there's less of a difference between these at every KD as for amplitude only. And if you look at the standard deviations, you can see the difference between these two plots implies what I call an information capture uncertainty principle, which basically means that there's an interference between capturing time information and amplitude information. Again, this is a prediction about how receptors and ligands might be designed or how they may have evolved to capture different kinds of information. So I created LR pair types, ligand receptor pair types from low affinity receptors and high affinity receptors in different waveforms, or based mostly on rise time. And what I found was that the rise time matters much more than the duration of the response. So you can see that even though these are very small responses due to the low affinity, you get a multiplicative effect at faster rise times. And then you can really compensate significantly more than several orders of magnitude when you go to a very high rise time of your waveform, of your neurotransmitter waveform at low affinity receptors, whereas at high affinity receptors, it really doesn't matter. It doesn't matter what the waveform is, it doesn't matter what the rise time is, you're going to get the same response. So now let's look next at the second messenger cascade dynamics that are stimulated by the ligand receptor interactions. So as a bit of background to understand this, I previously published last year a paper introducing the concept of a ligand pulse, which is basically an action potential for a biochemical network. So here we see a lactate action potential or ligand pulse. And one of the features of this kind of phenomenon is a phase transition. So you can see the shape of this hysteresis curve here, and it makes a phase transition to a completely different shape, which implies a different energy state. And this can also be seen when you plot the proximal metabolites. So in a cascade, here's pyruvate versus lactate, NADH, and ATP, which are the downstream options for pyruvate. And you can see that for low affinity receptors, there's a huge difference between the kind of hysteresis curves or states you get between fast transmission and slow transmission. But there's almost no difference between fast transmission and slow transmission when the receptor affinity is very high. So low affinity receptors better transmit information about neurotransmitter timing to the phase pulses or the ligand pulses. So this is an important sequence in this language that you can get information transfer from the ligand receptor to these state transitions or biochemical network states within the cell. So that one of the next important things to be able to interpret is the duration of the response. And so here I'm just looking at the cyclic AMP response, which is the immediate second messenger after the receptor activation. And I'm looking at pyruvate as a case endpoint. And so again, at low affinity receptors, we see quite a difference in the ligand pulse or the pyruvate action potential. But we don't see any difference at high affinity receptors. Oops. Sorry. And then we can see that the difference between a duration of X versus 5X, which is duration of the response, you see a very significant difference at low affinity receptors, but you don't see any difference in the phase plots for high affinity receptors. So only low affinity receptors transmit information about stimulus duration to the cascade metabolites, which then respond with phase transitions. So the next step is to show that these phase transmissions can communicate information to the effector site. And here we can see that the lactate ligand pulse, or the lactate action potential, is very different for short and long durations at low affinity receptors. And in fact, this is transmitted to the transient of the lactate response itself. And this is exported lactate. So you can see a very gradual increase at low affinity receptors, also a very graded increase as you'd expect. Actually, no phase transition here. At longer durations, again, this information then changes. You get a phase transition, which can be seen again in the lactate transient, where you get actually a response ceiling, which is achieved, which corresponds to this shape change. At high affinity receptors, you see less of a communication of this information, but you do see a different kind of information. You see that changing the duration can change the point at which the phase transition is made. Here at short durations, you get a phase transition even in the lactate transient, and the emergence of a secondary response, which corresponds to the shape change in the lactate action potential. And then here you can see that the shape of the lactate action potential is all that's really important in terms of the response of the lactate transient, and the actual amplitude of the action potential is not that important, or the lactate action potential is not that important. So the summary of the model can be seen in this schematic diagram, where you have any given ligand which can interact with the receptor with a variety of affinities, which then generates a second messenger amplification system and stimulates various metabolic states. So in the past, it was thought that you just get a second messenger cascade, and you get basically no difference or no information content in the state of the biochemical network. But I think this prediction shows that this opens up the possibility for a tremendous amount of information processing and bandwidth for computations in single cells, which might have tremendous implications for how neural networks process information. And this is just the model examples that I used that correspond to these steps. I would like to thank the entire molecular systems group and Professor Pierre Magistretti at KAUST for helpful discussions, and the support of the Blue Brain Project, the Swiss government, and KAUST. And now I can take your questions.
Thank you very much, Jay. Very, very interesting talk. Is there any question from the audience? Also for Eleftherios, maybe? Yes, Felipe, please.
I have a question for Jay. A fascinating idea. Thank you. I think I might have an example of of that idea at work. You know, one very mysterious thing we found in neurons is that the mitochondria respond with the, say, threefold activation with a very slow time course, well, relatively slow, within seconds, and it's up within, say, 20 seconds. But the feed-forward signal we think is important for that activation is cytosolic calcium. And so cytosolic calcium the transient is over in 3 to 5 seconds. So we have this delay between the signal and the actual output, and we've been really desperately trying to find the signaling cascade that would explain that delay. But according, correct me if I'm wrong, according to what you say, maybe we don't need it's just that maybe there is a very high affinity calcium receptor involved that explains the delay. Would that work for you?
Yeah, that could work. I'd have to know a little bit more about your system and your measurements, but it definitely could be a possibility. In fact, I would love to find some sort of experimental evidence or some real biological system that I could test these predictions in. But it is possible, because even though I was looking at ligand receptor at the surface of the cell, obviously at mitochondria, you have another membrane, you have another surface, you have a lot of interactions. And so there's a lot of amplification and there's a lot of information processing depending upon, I think the point is the state of the system. So you can have a biochemical network in a high state or a low state. I think this is what one of the major predictions of this model is. And so if it's in a high state, you could actually see perhaps a delay in the signal, or because you have a larger signal, you have a longer time of decay of the signal. I'm not really sure what you're measuring or when you're measuring it, but these kinds of processes could be involved in what you're observing.
Yeah, maybe if you want, I could write to you and to show you the data.
Absolutely, we'd love to see it.
Great.
It could be a collaboration.
I think I can conclude this session. So I want to thank all the speakers for their wonderful talks, and my personal acknowledgement also to this collaborative project for, I mean, all the opportunities that I had. And I mean, thanks to this experience, I could also progress with my career. So I want to express also my gratitude to KAUST, of course, and to this very rich environment. And thank you all for the attention. So I would leave the ball to Dan at this point.
Great. Yes, thank you very much. I'll give a little summary and wrap up. So what have we seen today? Well, we've seen an integrated workflow spread between two different institutions. In the first part, we obtain electron microscopy data. We reconstruct the surface of astrocytes, the endoplasmic reticulum, mitochondria within them, also the distribution of glycogen granules within these cells. To do so, we've had to develop new ways of segmenting the stacks, as well as visualizing the information contained within them. Using this data, we've been able to construct statistical morphologies that exactly replicate the characteristics of the astrocytes seen from the experimental data. We've then populated an entire neocortical microcircuit with these astrocytes and vasculature, and used then this system to run simulations and understand better the traditional interplay between metabolism and neocortical microcircuit activity. This has also allowed us to explore more theoretical approaches to understanding the signaling cascades within astrocytes. And finally, we've had to develop new methods to repair the meshes and visualize the results at scale. That is to say, when you have these massive systems with many, many different polygons and much complexity within the scenes. So I would say that, overall, when all is said and done, we'll have probably more than 20 papers come out of this incredibly fruitful collaboration. But moreover, what this collaboration has allowed us to do is extend the blue brain simulations to be beyond just neurons, right? for the first time, we have an integrated model of the entirety of tissue. That's to say, with glial cells represented, with the vasculature represented. And this will open the door to an exciting new future of being able to understand how neocortical activity can affect many of the more abstract measurements, such as MRI and blood flow that have been also collected throughout the years. So I also want to say that most of the data will be shared via the Neuronal-Glial-Vascular portal that will be available from the Blue Brain website coming soon. And I'd like to conclude by thanking KAUST, all our KAUST collaborators, including, of course, Professor Magistretti, and the Blue Brain, Professor Sherman, of course. I'd like to thank all of the speakers that we've had today. And finally, to conclude, I'd like to thank you, the audience, for attending this workshop. So I think we'll conclude the workshop here. Thank you so much.